All right, thank you to our speakers. We have a few minutes for some discussion, so I'd like to open up the floor for any questions.
Patty Gans, UCLA. Thank you both for your presentations. Federico, I have a question. You know, I've been running the cancer genetics program at UCLA for about 15 years. And now with all the panels that are out there, this is my frame of reference because I've been embedded in this for quite a while. With the Supreme Court decision, it's been wonderful that this has made it possible to have the testing available at a lower cost for patients. But I think there's a big problem with so many labs now offering these tests. And as Bill Gradishar said, NCCN is agnostic on what platform or whatever. How are we to know if a CLIA certified lab is now offering this test that particular, say it's a BRCA1 or 2 testing platform or a panel that's breast cancer, how do we know as the oncology consumer who's ordering on that test that it's really up to snuff? And I think, has that particular laboratory diagnostic test actually undergone review, or is it just, we've grandfathered in the lab because it's a CLIA approved lab? So I'd like to really know, and this really then goes to the next gen sequencing or whatever else is going to go on with the testing, because most oncologists, we're just used to ordering a CEA or PSA. We don't know how the lab director has chosen the particular methodology. So how can the buyer beware, if you will?
That's a really great question. And the CLIA regulations mandate us to validate a test, before we put it in clinical use. Now, the standards to which that test is validated are up to, at this point, the criteria of the medical director of the laboratory. I do think that there has not been enough transparency in the laboratory industry to actually say, this is what I did, and that was basically the impetus to create the genetic testing registry, which the NIH put together, in which all of genetic laboratories actually now have provided information about their test, their validation data. So that's one resource that you can actually go to the GTR at NCBI and look at what the validation of the assay was performed by the laboratories. I think it is becoming more and more important for physicians to understand what type of the test the lab is performing and actually request that information, request the validation information and how was this actually validated, demonstrate that you actually have the expertise to do that, show me how you did it, and how well you understand this field. I think it is very important. It is maybe onerous right now, but I do think that there needs to be improvement in how do we share that information, so that you can have the assurance that the laboratory that you're using actually has that appropriate validation.
But I can just tell you, just again, in the area of breast cancer genetics, where we've had a long history of this, most doctors, most oncologists who are even interested and motivated, who are now doing this testing, and if we're going to have universal testing for BRCA1 and 2, they don't know. And I can just tell you, the companies detail them. My genetic counselors are getting hounded by all of the companies that are doing the test. They don't have the time to investigate, the quality. And so this is a problem in an area where we're looking at germline issues. What is it going to be like when we get to the complications of the tumor genetics and, all of the interpretation, what's the bioinformatics behind it and so forth. So if there's any lesson that I've learned now just with this, year or two of having competition, if you will, in this field, it's very challenging. The end line oncologist who we're talking about doesn't even know how to handle the hereditary genetics, is not going to be able to do this. So I think that some quality control, some standards, there may have to be evaluations of the testing platforms, et cetera, because the average oncology consumer is not going to be able to make those discriminations. And that's just one of my concerns about the potential marketing of all of these tests directly to the oncologist who's at the bedside with the patient.
Yeah, I think that's a valid concern, but it is no different that, if you need to refer your patient to a physical therapy or to a palliative care, you need to figure out which of those providers is going to actually do the best for your patient. So yes, actually, I believe that increased competition is actually better because it pushes everybody towards better quality. And we do need to define, better standards. And more importantly, how do we share that information about, how the performance of the tests are in each laboratory?
Hi, Barb Conley, NCI. Maybe this question is for Karen, I think. When you are thinking about your business model and how you're going to cover the cost of the assay validation for companion diagnostics, and you're looking at this total life cycle review, how do you figure in LDTs that will analyze the same analyte that your companion diagnostic analyze?
We actually don't take them into consideration at all. And the reason being, the only time we would is if in the companion diagnostics there was an LDT that was used in the preclinical before we start the clinical series. We would look at the LDT to see how the primer probe was selected and if it's, and then look at what we're doing. But in general, we don't. We follow the requirements that we have, that we know the product has to fulfill, not only clinical validity, but clinical utility, and that's what we take through.
Okay, I just want to follow up that because what we hear a lot is that, you make a companion diagnostic and then the hospital lab or reference labs do a completely different test and they're testing that analyte and they use that even though the label doesn't say use a test that's not FDA approved or cleared.
We hear the
Still,
same
it
thing.
happens, and
we hear the same thing. Absolutely,
yeah.
we hear the same thing. And they get reimbursed.
And they get reimbursed, so that's not coming to Abbott to offset any costs of the ongoing surveillance.
No. Each institution makes that decision independently. As I said on my slide, the US FDA has made it very clear that when there's a companion diagnostic, there will be an approved product to go along with that. So, and that's the model that we follow. I think the bottom line is, regardless, we want the high quality product, wherever it's coming from. So that level playing field, but we will always follow the model of an approved product for our companion diagnostics.
I have a question if no one else, did you have a question? Go ahead.
I have a question. So my question is a corollary to Patty's question about performance standards. Sorry, I'm Deb Schrack from Dana-Farber. And it has to do with reporting standards. And I guess it's a question to you about whether you've thought about, there are all these companies now in the space of doing panel testing. Have you thought about achieving some consistency in your reporting standards, or are you looking to leadership from organizations represented in this room, whether the NCI, ASCO, IOM, AACR, to do that? And just sort of as an example, clinicians look and process lots of information, but every doctor in the United States picks up a lab report for a CBC, and what's at the top? The white count. And they know where to find the white count, the hemoglobin, and the platelet count. And it's the same. Lots of labs do it. They're all CLIA certified. But there's a consistency to how a CBC comes out or how a Chem 20 comes out that makes it possible for people to process information. When we get these panel results, one firm has tier 1, 2, 3, actionable, yes, no, maybe, Roman numerals, A, B, C, D. The taxonomy, if you will, is all over the map. And I guess just to move us forward and to help deal with this massive job of educate or engaging everyone, who's going to take ownership of taxonomy? Can you guys do it together? Can you guys collaborate across commercial entities? Or do you think we need to do it? And then if we need to do it, are we going to come together as a community and get IOM, ASCO, ACR, who am I leaving out? NCI, AMP-CAP, American College of Human, FBI, CIA, So I guess it just feels like, Patty's question is about the performance characteristics of the test and making sure that that's valid. That's one piece. But the other is simply, is this alphabetized from A to Z or Z to A? Because it can take half an hour because there's so many different places. And I know where to find a platelet count, but I don't always know where to find a BRAF mutation.
So, I mean, I think we are talking about apple and oranges, right? So the type of data, the very discrete data that comes on a CBC or those are easily, arranged in a table format, and they've been used for, more than 50 years, that we use those formats. So there's been time, right? We are using panels now for, a few years. And so we're still all learning. Having said that, there are standards for reporting. Both ACMG and CAP have standards for reporting of the type of information that needs to go into the report. There are no standards for the organization of the information. It's just standards for what information needs to be there, right? There's definitely, from the germline side of things, there's very clear standards of how to categorize variants, mutations, what do they mean, and what information to actually put and how to put it in the report. So that is relatively fairly, I would say, standardized. On the somatic side, there's nothing, right? And as I think was discussed before, the term actionable is all over the map, right? All over the map. Anybody can call actionable whatever they feel they can call actionable, as long as there's some action that's going to happen afterwards, whether that action actually benefits the patient or not. So that, I think, is the crux of the matter. And I would say that it's probably going to be on the side of the organizations, ASCO, AACR, CAP, and all of those, to get together and come out with standards of what can you actually call actionable, and that I actually that we support as an organization, because I don't think many of the companies are going to want to agree on that, because it's part of how they use the information, right? I know there's companies who say, well, 90% of our gene panels have, actionable results, and that again depends on how do you define actionable.
So I will just add to your point. I was told that Oncotype DX, when they developed their test, which was a very different gene signature, that they went through over 80 different formats for their report before they finally, and did usability testing and all of the stuff before they settled in on the report that we now see. And so we're still very early on, and this is actually an active area of research, and not just in the hands of the molecular labs. It's an actual area of how do we best deliver this type of information, and we are so early in that game that and there's so many players now, not just one company, Oncotype DX, doing usability studies. So I think we're going to see a lot of variability in how we report these, and hopefully there will be organizations, I think one was mentioned yesterday, coming together to try to come up with these types of standards.
and believe me, the variability on what you perceive to be a a good report versus what Mia perceives to be a good report, it's all over the map. I can tell you, we redesign our report, we send it out, and we get calls saying this is fantastic, we love it, and then some other people call and say, what did you do? Why did you change this? We don't like it. So there's also a lot of consumer difference and how people like to see that information.
I have a question. I want to bring together two thoughts that have been expressed here today. One is this idea of how we don't want to cut off innovation by making things too difficult to develop these tests and get them out to market. The other was that it's very difficult, particularly for academic investigators, to take those final necessary steps to really optimize an assay and get it ready for clinical use. So as people who are involved in doing that kind of development work, what advice could you give to academic investigators so that if they develop a test, they would be best positioned to approach you and have you agree to do those further development steps?
We actually have probably several hundred, depending on the disease indication that we're looking at, whether it be academia or researchers, in which and not only outside of the pharmaceutical, but inside the pharmaceutical companies as well, that we have focal panels, advisory panels all the time, just to make sure we are gathering that information, because that is important. It helps us move through our process uneventful, which is always the direction you want to take. I think one other thing and I'm going to add to that, to make it, as I look at what we're doing within the United States, and the FDA has recently agreed that this is the path that we're going to try. There's going to be a pilot program. We want to be able to get the assay out there, but remember, we have to either do it through an investigational device exemption, or in rare disease cases like you're talking about, those clinical studies are going to be quite lengthy and quite long. So there's actually a model that's being developed in which there will be assays that will be able to go to market while they're collecting this clinical utility validity data, and then finish the FDA filing. This is very, very early, but that's another path that is being developed and evaluated by both industry and FDA as a pilot program, that's another path that we can do that as well in working with our academia and key opinion leaders as well.
I would say that, putting on my academic hat, I think we're not necessarily trained, even as laboratorians, to understand the process of commercialization of a test. And I would say that for those that are in academic institutions, the best that they can do is get themselves familiar with their technology development offices, and who are always very familiar of how to bring the team together and how to identify the best partners for actually getting that test developed into a commercially viable diagnostic. So I think that intellectual property management or technology transfer offices are usually fairly helpful in trying to move a diagnostic forward. And they already have, most of them, the relationships with the industry that need to happen. So the message is you don't have to do it alone, right? It's like there is a lot of opportunity out there, and just reach out to those groups, and they will find you the help that you need if the diagnostic is promising.
Hi, Suanna Brunigi with ASCO. I'm wondering if you can comment on the parallel review pathway with FDA and CMS, and what you think about that as a mechanism and whether that, having that sort of combined process gives the companies the incentive to produce the data that ultimately may demonstrate clinical utility.
There was a pilot program for that exact review, and actually Exact Science, I believe, was the first one that went through that parallel. It worked very well for them. I think from an industry perspective, CMS is going to have to get involved sooner. They've been a bit reluctant in the past, and industry's been a bit reluctant to have them included in the past. And the sheer reason was the conversation evolved. Well, you can't evolve. You have to all agree in the very beginning what those studies need to look like from a reimbursement perspective. And that gets added on to what we would do from a registration perspective. There are some similarities, but there's absolutely additional information that gets collected for the CMS assessment. So you're absolutely correct. We've seen it work now once. I think it will be more common moving forward now that it seems to be the model starting to shake out as to how that will work. It'd be the best way to do it, obviously, instead of waiting till after. US isn't the only one that does it after. In Japan, you go through the complete process before reimbursement can start. That's just the way their laws and regulations are set. Same thing in China. So you go through the entire process, then you file for reimbursement in Japan. So it's nice that we're looking to do it in parallel. I think that will help.
Okay, if that's the end of the questions, I guess we'll conclude our session.